that program, Emory, Georgia Tech.
Yeah, and he and I are serving on one of the AICHE program committee. So I think I saw him perhaps last week or at least before, virtually. But
I see. Small world, I guess. So maybe I get started. Good morning, everyone. Welcome to another Terasaki seminar series. Today we have Professor Ankur Singh. He's a Woodruff Faculty Fellow and Associate Professor of George Woodruff School of Mechanical Engineering at Georgia Institute of Technology and Wallace H. Coulter Department of Biomedical Engineering at Georgia Tech and Emory University. Before Georgia Tech, he was an Associate Professor at Cornell, where he served on the Center for Immunology's Executive Council and as the Associate Director of Cornell's NIH Immunoengineering T32 Training Grant. His laboratory works on immune organoids and enabling technologies to understand healthy and diseased immune cells and translate therapeutics. He's received many funding from federal agencies, including National Institute of Health, NSF, Wellcome Leap Hope, DOD, DITRA, Lymphoma and Leukemia Society. Published more than 60 papers in top journals, including Nature Methods, Nature Materials, Nature Nanotechnology, Nature Com, PNAS, Advanced Materials. He's also written multiple editorials for Science Translational Medicine. He's won many awards. He's a fellow of AIMBE, recipient of the prestigious NSF Career Award, Society for Biomaterials Young Investigator Award, CMBE Young Innovator Award, CMBE Rising Star Award, DOD Career Award, etc. The floor is yours, sir.
okay. All right. Thanks, Mehmet, for the invite, and thanks, Ali, for the invite as well. And I'm excited to present some of the work that we have been doing for the past several years at Cornell and now at Georgia Tech, as we have continued in some very exciting directions from here. In my lab, we study several important areas, including infectious disease, inflammation, cancer, specific cancers such as lymphomas and prostate cancer, and also aspects of gut microbiome. So I'll be able to show some of these things today in a more summarized form. But in general, what we do is we go from biomaterials engineering to technology development and then use that technology to study mechanism or develop new therapies, which then hopefully can be translated towards treatment of those diseases. One of the central piece to the lab is the immune organ, and that constitutes about 2/3 of the talk today, and then the last part will be a newer work from the lab, which is in the area of prostate cancer organoid development using hydrogel-based materials. So what you see on the screen is a snapshot of a human lymph node, and these are, you have about 450, 500 of these lymph nodes that are strategically placed throughout the body to sample and prevent us from infection, and also are engaged in vaccine responses, as many of us are now familiar with this over the past couple of years since the pandemic had started, how our immune system works. But if you take a cross-section of this, what you're seeing is a nicely formed these green structures. These are B cell zones. These are one of the type of immune cells found in our body. And then the red are T cells, which are another type of lymphocyte immune cell found in the body. And surrounded by those are these collagen-based meshes that you see in yellow color over here, which also infiltrates into these pockets that are T cell and B cell zones over here. But what you're really seeing is a snapshot picture. For most part, if you Google, you will see a lot of these histological or immunohistochemistry, immunofluorescence images, which are snapshot pictures that tells you that how compartmentalized, well-decorated these lymph node structures are. In reality, these static structures are highly mobile. So in a study that we published in collaboration with Prof at Cornell recently, we did 3-photon imaging to look at these highly mobile immune cells that you're seeing as T cells, as red and green of cells with subtypes of T cells, and they are really going back and forth, up and down. And you have these vascular networks in the surrounding, which can be lymphatic vascular network or blood vessels and things like that. So this is a T cell system. Let's go and take a look at a B cell system. Okay. So here you're seeing another complex structure within that B cell follicle that I mentioned, which is the green zone that I showed early on. These B cells are, each of these colors that you're seeing is a particular competitive clone that's trying to win the race and become the antibody producing cell. And as you can see, each of these are randomly moving around, trying to find better connections, trying to make certain decisions. And all I'm trying to say is that this is a very mobile structure, highly motile cells, which you don't see when you're looking at the snapshot of a lymph node or a spleen or other lymphoid organs that are involved in the immune responses. So how could we build something where you can do these dynamic reactions outside of the body? And that's where my last research has focused on building these engineered lymph nodes ex vivo or lymphoid tissues. Primary reasons being for us to understand the decision making in these cells, what makes them start, what makes them stop, what makes them conclude or makes, for example, antibodies or react to various infectious agents. Can we develop immune therapeutics in a dish? So instead of using hybridoma models or by using animal-based models, can we make immune therapeutics in a dish, for example, antibodies? And then how all of these immune cells converge into malignancies and disorders that are related to things that originate in the lymph nodes or things that migrate to the lymph nodes. So for example, lymphomas are cancers that originate in these lymphoid tissues, but there are a lot of other solid tumors that might invade into or metastasize into lymph nodes. So can we use these ex vivo platform to study some of those things? So now I'll take you one step further into the B cell immunology and to educate you on how the system really works in our body. So when we get infected or vaccinated, then okay, my mouse is here, okay. So the antigen or the infectious agent or the vaccine component goes into the subcapsular sinus of the lymph node. From there, next to that is this B cell follicle, which if you recall was the green compartment that I showed in my first slide. The information in this transported into this through various means. And at this point, several cell-cell interactions and cell-cell interactions become very important. So one of those is the T cell interaction with the B cell interaction by means of a receptor ligand called CD40L ligand interacting with the CD40 on it. This primes the B cells to move forward in the decision-making process. One of those decisions is just become a short-lived plasma cell and become a low-affinity cell, which is not very desirable. The second is undergo this complex reaction called germinal centers, which are transient structures that originally do not exist, but when you are exposed to infection or vaccinated, these structures appear. In this, the B cell undergoes massive proliferation followed by diversification. So the repertoire has to reorganize, in simple words, so that it can be specific against a particular infectious agent or an antigen or a protein. This happens in a zone called dark zone because it's optically dense, and then it migrates under the effect of chemokines into the next compartment called light zone, where the optical density is less because there are less number of cells in this area. But this is an important zone that allows for the cells to undergo decision making. So they really interact with these specialized follicular dendritic cells and specialized T cells to ask whether they are good to move forward or should they be eliminated. If they're good, they undergo this whole process again and again, multiple times, and very much like a Darwinian approach where there are many competitors, in the end, you are left with a few clones that are becoming the memory and the plasma cells, which eventually save us. So how can we recapitulate this very complex process outside of the body? We went back into the classic approaches that have been reported in the field. And one of those was developed by Daisuke Kitamura in Japan, where they took a fibroblast line to transduce it with the CD40 ligand. And when you take a naive B cells and co-culture them in a two-dimensional co-culture system under the effect of cytokines, you should induce some level of germinal center B cell maturation response. Well, when you do that, you have very few cells that actually undergo that reaction, and they are short-lived cells. When we performed some extensive sequencing analysis, we found that even though these two-dimensional cells were undergoing, some of these cells were forming, they were not really close to what an in vivo immunized mouse-derived germinal center B cells will take. So, for example, EGDA is a gene which is critically involved in germinal center formation. In a naive B cell, it is not present. In an in vivo, it is highly expressed, but in a 2D system, it is not expressed to that similar level. Likewise, there are other genes that are up and down, but not really mapping to what in vivo system does. So clearly, this system is not working very well. So we took a step back to understand what's in the microenvironment of these lymphoid tissues. So in one of the approaches, we collaborated with Jason Pfister at UCSF to look into some of his single cell sequencing work that he has done for the stromal population and combine that with the extracellular matrix components and integrins and other adhesive components that are found in the tissue microenvironment. For example, if you look at these various stromal cells that are found in the lymph node, identified by Jason's group using single cell sequencing, FDC represents follicular dendritic cells that are found in these B cell follicles. And VCAM1, collagen1, vitronectin, fibronectin, many of these important bioadhesive genes that represent bioadhesive proteins are highly present in these compartments or in these stromal cells. Same way, if you evaluate in specific extracellular matrix proteins, for collagen1 is present, vitronectin is present, VCAM1 is also presented by stromal cells and some other adhesive components. So collectively, we took that information and went on to engineer our first immune organoid several years back. So in this approach, we took the same stromal cells that I showed earlier. They are called 4DL cells. And then you combine that with a naive B cell, freshly isolated from the spleen of a mouse, for example, or lymph node. And then you co-culture this whole thing in a two-dimensional system. This two-dimensional system was formed by combining gelatin with silicate nanoparticles. This is a work pioneered by Ali's group, as well as later on followed by Akhilesh Geharwar's group. So we collaborated with Akhilesh on this back in 2014. And the advantage here is that by using these two-dimensional nanoparticles, you can cross-link these gelatin structures and prevent them from forming just a simple liquid. You can control the porosity of these gels by either changing the percentage amount of silicate nanoparticles or by changing the weight percentage of gelatin. And we also matched the storage modulus with some of the bulk properties, mechanical properties of the lymph node that we had quantified in collaboration with Jonathan Busher at Cornell by using a micropipette aspiration technique. As an outcome, when you take these optimized hydrogel platforms and you take single B cells from spleen and add to these hydrogels, within 3 days, these cells start forming these organotypic clusters. They self-assembled into these organotypic clusters. And the differentiating B cells that are shown in green with the GL7 marker are now co-localized with the spread fibroblast that is presenting the T cell signal. So imagine these fibroblasts as a T cell mimic that is communicating with these B cells, which have now undergone a conversion. Quantitatively, using flow cytometry, we found that it was increasing this whole reaction by about 100-fold compared to much lower expansion of this phenotype, which represents germinal centers, by using a two-dimensional system. And you can control this by changing the biomaterial property or by changing the seeding density of the 40L B cells that were presenting the CD40L signal. Now, this only represents a phenotype. It doesn't really tell you a lot of other critical hallmarks of germinal center reaction. To prove that, in collaboration with Wendy Begulin and Ari Melnick at Cornell Medicine, we performed these extensive benchmarking assays. So one of those was looking at the entire transcriptome of the immunized B cells, germinal center B cells from immunized mice, and compare that to organoid-derived germinal center B cells. So here's an germinal center B cells isolated 10 days after immunization of a mouse, taking the spleen out and isolating these cells and comparing that to organoid-derived B cells, and they're remarkably close to in vivo germinal center B cells. So this was one of the earliest findings from our group, and perhaps reported that you can develop a hydrogel-based ex vivo system that generates germinal center B cells that are so close to in vivo germinal center B cells. Not only that, the second hallmark was somatic hypermutation, which is the VDJ recombination that represents your repertoire rearrangement, for example. And that was also pretty remarkably close between organoid-derived B cells and germinal center B cells. So likewise, we did a lot of extensive analysis and benchmarked really what kind of system we can do. And then these were the initial set of organoids that we used for a variety of questions moving forward, including understanding the antigen-specific response. But if I go back to the slide where I showed you have VCAM1, collagen, vitronectin, fibronectin, a lot of those parameters. Gelatin is, you cannot easily modify gelatin to present a lot of these factors. So you need a more modular system. So in generation 2 of these organoids, we moved into developing a PEG, 4-arm PEG maleimide system where these arms of maleimide could be functionalized with thiolated peptide ligands that undergo click chemistry reaction, a Michael type addition reaction. And you can form these cross-links by using dithiolated peptide cross-linkers, which you can design to be protease degradable or MMP, matrix metalloprotease specific degradation in these cases. So you take the same co-culture system and now put it in these designer matrices, and we compared that to the vinyl sulfone-based systems and acrylate-based systems that undergo similar reaction kinetics. They may have some other parameters that might influence certain characteristics. One of the most important findings for us was that the B cells really like the PEG maleimide matrix or hydrogel system, where these phenotypes were developing very well as compared to the vinyl sulfones and as well as the acrylate-based system. Okay, so now that we have a designer system where we can simply plug and play with peptides of interest, so we asked a question about, let's take a collagen mimic Gefolder peptide, or take a fibronectin mimic RGD peptide, or a VCAM1 mimic or EDV peptide, and functionalize and look at specific molecules, proteins that represents aspects of germinal center reaction. And by this data, I want to emphasize on how this kind of designer system enables you to perform time-dependent studies with different matrix combinations, various parameters. You can practically, you can theoretically functionalize with any protein or peptide ligand of interest as long as it's iolated, with some tweaks as necessary for the hydrogel formation. And as you can see with the Gefolder system, you're seeing a very rapid increase by day 4 and a sudden decrease by day 6, which kind of starts telling you that the B cells are maturating by day 4 and then rapidly exit the system towards the next stage. In contrast, if you take VCAM1 mimic, you have a pretty stable 4 to 6 day response, suggesting that this particular peptide may have a delayed exit later on. So these kind of questions could be asked, which are not easily answerable by using a mouse-based system that are historically used in the immunology community. So we went on to test this to understand the response of predicting B cell immunity in against bacterial antigens. We also focused on aging as a parameter, for example. And The rationale was that the antibiotic-resistant bacteria, they're present a major threat globally, and of these, a particular type called Klebsiella, it is of high interest because it's an antibiotic-resistant bacteria which causes a lot of ICU-based infections. And unfortunately, this bacteria, it fails to induce in vivo germinal center B cell responses. So you don't really get very highly effective antibody responses. And because this is antibiotic resistant, so you need alternative therapies. So vaccines and antibody-based immunotherapies are emerging as important area for these treatments. And what we wanted to see was, can we develop a system where we can study these Klebsiella bacterial components and determine how to better build vaccines, which component to take from a bacteria that will lead to a better germinal center response and better develop or generation of antibodies, which can then hopefully be collected and infused back into the patient. So to do that, we collaborated with Ilana Brito at Cornell. She's a microbiome expert. And with her help, we were able to generate from this Klebsiella pneumoniae an outer membrane protein that had these decorated portions, and then a purified soluble OMP as a comparison. And when you take that and when you make these organoids and you incubate simultaneously during the formation of these organoids, these specific antigen components at predetermined concentration, you can look at how they are inducing not only the phenotype of germinal centers, not only the gene expression, but also downstream epigenetic changes. For example, histone trimethylation is a hallmark. In coming slides, I'll show you how important it is. But this is a very important step that allows for germinal center reaction to proceed. So the question is whether the outer membrane protein impacts trimethylation more than soluble or the other way around. And if we can understand that, we can better design these vaccines to either have outer membrane protein or soluble components and lead to more Klebsiella, treatment of Klebsiella-based infection. As you can see, you have a significantly higher shift in the trimethylation of histone by using soluble OMP as compared to the outer membrane protein, as an example. We further use this to study the differences between aged versus young germinal center B cells. To give you a little bit of information, as we all understand, aged people don't respond very effectively to infectious agents. And there are information on whether the B cells are undergoing immunosenescence or the T cells are not expressing the right set of molecules such as CD40 ligand to induce B cell differentiation. So we thought if we take B cells from both young and aged and give them an opportunity to differentiate by forcibly presenting the CD40 ligand using these lines in the two-dimensional matrix, then can we first generate aged germinal center B cells, which you cannot generate effectively in an aged mouse by immunization. And when we do that, indeed, we can induce germinal center reaction in the aged mice, however, to a lesser extent than young mice, which could be tied to some level of incapabilities of these B cells in their naive form to respond to the T cell signals. Then what happens if you take these cells aged versus histone trimethylation? And that also indicates that the trimethylation of histone in these aged B cells in the organoid was there, but it was less than young. So the conclusion is that by taking aged population, we are able to generate these germinal center B cells, which is one of the first reports, I believe. But then we're also able to determine the difference between the young and the aged population. So we're very excited to take this forward and more thoroughly study the aging effects in the B cell area. Now, what happens when you take these Klebsiella components and add to these 8 samples? Here you can also see that by using soluble OMP, you're getting much more trimethylation as compared to Klebsiella outer membrane protein, very much like what you're getting with the young population, suggesting that if you can make these 8 organoids by adding soluble OMP or Klebsiella, you can generate germinal center B cells, which can hopefully then generate antibodies that can be transferred back into the patient for treatment purposes. Okay, I'm going to shift a little bit of gear and take you to the next level over here. Mehmet, just please give me 10-minute timer, like a heads up when I'm close to that.
Okay.
In B cell follicles, we've spent about 8-9 years on one compartment of the lymph node. It is so complex. One of the complexity comes from the follicular dendritic cells. These are interesting cells that capture antigen and just grab and present it to the B cells. So if you add a soluble antigen to the organoid, that soluble antigen is just going to bind to the B cell receptor, but there is no cell-cell interaction between a follicular dendritic cell and a B cell. So we hypothesized that in vivo, what happens is that from the follicular dendritic cell surface, the antigen is grabbed by the B cell, it clusters, and then this fragment of follicular dendritic cell is extracted along with the antigen for B cells to undergo maturation. So can we reproduce this aspect of membrane, the fluidic membrane, by using a lipid-based particle that presents the antigen in contrast to a soluble antigen? And this work is done in collaboration with Prof at Georgia Tech, led by Leanna Kramer in lab, PhD student. And so we formed these liposomes that were made from lipid components, and we characterized them in terms of their loading efficiency. And then when you incubate it with the B cells that are undergoing germinal center differentiation, and you take a look at the unloaded liposome versus an antigen mimic loaded liposome. So we use anti-IgM as an antigen mimic, and the liposome surface was coated with the covalently conjugated with these anti-IgM as an antigen mimic. You have a far more take with the B cells of these antigen loaded liposome compared to the unloaded liposome. Now, what is the downstream effect of that? This particular phenotype represents the maturation of B cells. And if you compare soluble antigen versus liposome-bound antigen, you have significantly higher induction of germinal center maturation by having a membrane-bound presentation of antigen. So this emphasizes the importance of engineering these kind of systems that allow to get you a step closer to what might be happening in vivo as compared to simply using approaches such as soluble antigen and likewise the system. And to investigate further how this might be helping, we looked into some microscopy of at the single cell level of the B cell receptors and found that the cluster area of these B cell receptors was significantly higher with the liposome compared to the soluble. So the cartoon image that I was showing a couple of slides back was that these antigens are bound and then B cells undergo clustering. With soluble, you're not getting this clustering, and this clustering is critical for the downstream signaling inside the B cells, which ultimately leads to the formation of sector cell. Okay. So if I've convinced you that these approaches, engineered approaches, are pretty impactful for studying some of this, one of the ideas we had was to answer a fundamental question in immunology, that when you get infected or vaccinated, your germinal center B cells massively proliferate. The pioneering work in the field has shown that that happens. But what controls this pace? How can you understand or figure out which proteins are involved in this? And if you can understand that, then you can use that information to develop better antigen and adjuvant design, make better vaccines. And so to answer this question, we looked into some of the epigenetic molecules based on some prior information with our collaborator, R.M. Melnick. So these epigenetic molecules, which are either undergoing methylation or doing methylation processes or acetylation processes, there are several of those that are important in the B cell maturation. One of those is EZH2, which is a histone methyltransferase with a very long name over here. Many of these molecules that you have listed over here, they are in on or off state. Not everything is on, not everything is off. So they're in different states and critical for the process. So based on some analysis of primary human tonsil B cells with chip sequencing and some existing prior art from our Melnick's work, we came up with a hypothesis that EZH2, the histone methyltransferase, whose job is to trimethylate histone 3K27, is a master regulator of the striking germinal center proliferative phenotype, and it does it by suppressing or repressing cell cycle checkpoint gene CDKN1A. To study this, test this hypothesis, we initially invested a lot of effort into using mouse model. So we have a wild type CDKN1A knockout, EZH2 knockout, and a cross of these two, which is a double knockout of CDKN1A and EZH2. And if you just focus on this particular panel shown over here, you can see my mouse. You have, the second row. You have nice germinal center formation in wild type CDKN1A, but no germinal center in EZH2 knockout. And when you have a cross of these two, then you rescue the germinal center response. This suggests that if we lose EZH2 for a variety of reasons, just so you know, EZH2 is becoming popular treatment for certain medications, certain diseases, cancers and all. But if you use EZH2, you also suppress your ability to induce germinal center reactions. So you're not forming germinal centers over here, but clearly there's a crosstalk between EZH2 and CDKN1A, which we needed to understand. So to understand that, we went back to our Organoid platform, and we first asked, can we study EZH2 in the system? And if you take a look at germinal center B cells in vivo from immunized mice, they have EZH2 expression, and the organoid derived germinal center B cells, they have EZH2 expression. But the two-dimensional cultures of those 40LB cells do not express EZH2 gene. Likewise, many other genes for up and down that are critical to the process. So one of our most important findings over here is that now by taking freshly isolated naive B cells from the spleen of these 4 mice that I explained, wild type, single knockout, and the double knockout, we can really recapitulate the germinal center reaction ex vivo in a dish similar to the in vivo mouse. So here is the in vivo response, and you can compare that to the ex vivo on dish response, which is remarkably similar with much higher output in a controlled manner that it has. So it shows the power of, again, an immunoengineered system now that is capable of recapitulating aspects that were not previously shown to have been successfully demonstrated. Long story short, by using this, we were able to, this work was led by Wendy Vergulen, she's a professor at Cornell Medicine now, where B cell and T cell undergo interaction in response to an infection. They go uphill, and at the top of the peak, they have, and there is an interaction between EZH2 and CDKN1A, and when you take these breaks off, cells undergo rapid proliferation in our body, and this cycle continues until we have a winner population, which is the plasma and the memory cells that are formed. And then we, using various transcription factor analysis and some bone marrow transplantation analysis, and likewise, Wendy was able to identify that there is a cell cycle forward loop that's listed over here that drives the germinal center reaction through this unrestricted transition. So there's a cell cycle transition called G1S that was critical in that. So, you know, there's a lot of deep immunology and biology involved in this, but what I want to emphasize is that by building a system, we were able to do this. Without that, it was incredibly difficult to answer any of these questions because those models, the mouse models, have serious limitations of not being able to give us the kind of control information that we want from this. Okay, so now I want to shift gear and get into more on the Germinal Center side, but now from a materials-based vaccine development, and introduce a concept of gut microbiome. And some of the recent studies have shown that the vaccine responses are very dependent on gut microbiome. These are conventional, for example, influenza vaccine that we get, seasonal influenza vaccine. And this is controlled by secretion of metabolites, secondary bile acids that regulate the B cell responses. So we had a hypothesis that many engineered nanovaccines are being made or tested in labs, but we mostly test them on healthy wild type mice, but not really on systems that have an altered microbiome. If you look at the human data from Polymorphism's lab at Stanford and originally at Emory, there's a clear indication of how alteration in the microbiome, in the gut microbiome, impacts or fails the response of human influenza vaccines under certain conditions. So we asked the question if conventional nanovaccines such as PLGA-based vaccines will perform similarly under a healthy versus a more altered gut microbiome condition, and are there ways to overcome those restrictions? So here we looked into a model of disrupted gut microbiome by using TLR5 knockout mice. To give you a context, TLR5 is an important toll-like receptor protein present on the epithelial cells and many other cells in our gut system, and it senses the flagellin, which is the tail part of the bacteria in the gut, and it is required, this sensing is required for homeostasis in our body. When you disrupt that by using a TLR5 knockout, so you remove this TLR5, you still have the flagellin, but the body cannot sense it and kind of thinks that there is some abnormality. That leads to a condition called metabolic syndrome. Here in the TLR5 knockout, you develop this large fat pad mass, you have massive increase in large increases in weight of the body, as well as development of inflammatory signatures such as IL-6 secretion, leptin secretion, as well as collectively what happens is not only obesity is there, you have a diabetic situation developing, you have systemic inflammation. So it's very much of an umbrella term called metabolic syndrome that develops over here. So when you use wild type mice and inject with a soluble antigen, and you use a wild type PLGA vaccine in a wild type mice, you get a beautiful response in terms of the Germinal Centers. But when you use a soluble vaccine versus PLGA vaccine in a TLR5 knockout mice, you don't have Germinal Center formation. So clearly, disruption of the TLR5 sensing and induction of a gut-mediated metabolic syndrome was dissipating or eliminating the vaccine responses over here. Interestingly, when you inject the mice, after giving the booster, the vaccine also changes the gut microbiome. And this was surprising because we were thinking that it is the initial gut microbiome that is causing changes to the vaccine. But seems like there's a loop where initial is impacting the vaccine, but then the vaccine is further impacting the gut microbiome and the cycle continues, and that might be contributing to how these vaccines are not working in an altered microbiome condition. So these differences existed at the level of male versus female. So it was the male mice that did not form Germinal Center reactions. Female were just fine. They were forming it, partly because females in the time group that we studied did not develop the metabolic syndrome, but maybe at a later stage, they may develop some of these metabolic syndrome. Now, the key question is that PLGA vaccine is extensively studied, and it is not working in an altered gut microbiome condition. Can you overcome that by engineering a better system? So we engineered this modified polyhema system with pyridine, which introduces hydrophobic and hydrophilic domains. And when you bring a protein of interest, it self-assembles into a nanogel as a nano vaccine. And when you do that, and immunize, for example, let's go through this one more time. So TLR5 knockout with the PLGA vaccine has poor response compared to the wild-type PLGA system. When you take the PLGA with an adjuvant and use the same TLR5 knockout, you do rescue the response. So it seems like the adjuvant system is adding and bringing it back, but it's not enhancing it any further than the PLGA wild type alone. So, so really there is no major impact seen over here. Now, when you take the nanogel that's shown over here with the same antigen and without any additional adjuvant, both wild type and TLR5 are inducing superior response, germinal center response than this particular system. So this tells that by engineering an immunomodulatory material, you can stimulate B cells to undergo maturation and overcome the limitations that are imposed by altered microbiome-like conditions. Now the question is, how is the system even working? So we performed an analysis of which particular toll-like receptor these nanogels might be engaging, and it appears that they are engaging toll-like receptor 2 on the surface of these B cells as compared to toll-like receptor 5, 4, and 9. I'm happy to answer questions related to more details of this. So the model we propose is this, that when you have a healthy microbiome and you take the conventional nanovaccine such as PLGA vaccine, you get a nice antigen-specific germinal center response. When you take the conventional nanovaccine and have an altered microbiome, you reduce that response. You lose the antibody response. But if you engineer an immunomodulatory nanovaccines that can stimulate through various toll-like receptor pathways, then you can overcome these restrictions imposed by altered microbiome and induce better response. This is important because if you look around, most of us have different microbiomes. As you know, we eat differently, we live in a different environment, we travel and all of those things. In fact, we did certain studies with antibiotic treatment. If you wipe out the microbiome with antibiotic, or you change the composition of the microbiome with antibiotic, that fails the vaccine responses. So we have to be very careful with how we interpret vaccine responses just by simply taking healthy wild-type mice. And no, we should be looking more comprehensively into a variety of other biological factors and environmental factors that are happening. Mema, do I have more time?
10 more minutes, yeah.
Oh, I have 10 more minutes, okay. All right, so I'll now switch to my final part of the talk, which is a different topic, but it uses a similar biomaterial platform that I introduced you to you earlier, and also touches upon the topic of EZH2, the histone methyltransferase, the epigenetic system. So a couple of years back, we started looking into development of prostate cancer organoids because there are no synthetic organoid systems that are there. Only matrigel-based systems exist so far, and it's been one of the very challenging areas to develop organoid platforms. We were particularly interested in a progressive reprogramming of prostate cancer. So if you have androgen-dependent castration-resistant prostate cancer, eventually what happens is it acquires changes to in its system by means of epigenetic reprogramming by upregulation of EZH2 and downregulation of androgen receptor. So it stops these transformed tumors called neuroendocrine prostate tumors that are derived from castration-resistant prostate tumors. They lose their dependency on AR, which is the androgen receptor. And so the conventional treatments such as hormone therapies and all do not work on these things. As you can see in this gene expression analysis on a large number of patients, the NAPCs are losing their expression of AR. And if you look at these immunohistochemistry analysis, you will see that compared to a benign prostate, here is the castration-resistant prostate cancer, adenocarcinoma, and here is the CRPC-derived neuroendocrine prostate cancer. So this is the one that has transformed over the time, and this is the one that is expressing excessive amounts of EZH2. If you remember a couple of slides back, I talked about EZH2 as an epigenetic molecule. It's a histone methyltransferase. Its job, one of the goals is to take histone 3 and trimethylate it. So the question we had was, can we take these patient-derived tumors and grow them in a synthetic hydrogel platform? Prior to our attempt, the only system that existed for NEPCs was a matrigel system with our collaborator, Himisha Beltran at Dana-Farber. To understand that, we started looking into the tumor microenvironment of these prostate tumors. So we looked into patient-derived CRPCs, we combined multiple analysis techniques. For example, on the left, you are seeing RNA spatial omics showing distribution of various RNA of the collagen, EZH2. On the right, you're seeing collagen protein expression by using second harmonic generation in the tumor area. And we also performed analysis comparing various integrin types that might bind to this. When we take all of this information, there's a lot of information on the slide, and we combine with some proteomics information, what comes out is that collagen, fibronectin, and aspects of vitronectin are important for in these patient-derived tumors. So now that we have performed these RNA-seq analysis, mass spec analysis, we take the selective information and we go to the PEG-4 maleimide system and functionalize these arms with peptides that mimic these identified extracellular matrix components. We take patient-derived organoids that are first taken, so the patient tumors are taken, then first derived in matrigel for stability, and then from there transferred into the PEG system and cultured for several days to look at their progression. How do we design this? I mentioned that we take information from these, but we also look into what kind of cross-linkers to be used. So when you look at matrix metalloprotease 9, you have a significantly higher expression of MMP9, for example. So this cross-linker can be designed to be MMP9 specific or in general protease specific system. When you functionalize these hydrogels with Gafogar, which is a collagen mimic, REDB, which is a VCAM1 mimic or fibronectin kind of mimic, in RGD, which is vitronectin, fibronectin kind of mimic, and compare that to the matrigel, these microenvironment conditions lead to very distinct morphological changes. So you have these nice structures, hollow structures, tubular structures forming with matrigel, and then you have these pretty dramatic structures that are forming with collagen versus other matrices that are there. When you take this morphological analysis and now look into the expression of EZH2 across these matrices, so this is the designer system with 3 different matrices, compare that to matrigel. The EZH2 expression is dependent on which ECM matrix you're presenting. And the histone trimethylation also correlates with that, which ECM matrix you're presenting. For example, Gafogar is higher than matrigel, the collagen mimic is higher than matrigel, and REDV is higher than matrigel as well. And there are differences between RGD and REDV. So here's a cell, here's an extracellular matrix, there is an interaction outside, but our readouts is at the nucleus level. So how is this information even transferred from this interface of integrin extracellular matrix to the nucleus and leading to these changes in EZH2 chromatin level and histone trimethylation level? To study that, we performed some inhibition studies and eventually found that this information was being transferred to the actin cytoskeleton, where if you inhibit that interaction or add this the Y compound that is an inhibitor of the Rho kinase system, then you dramatically, significantly reduce the trimethylation of histone 3 in these prostate tumors that are there. We went on to perform these unbiased analysis of what pathways and how these tumors. I mean, one of the key questions is that we have these 4 different matrices. 3 different matrices? How do they compare to matrigel, and how do they compare to the patient system? And the take home is that each of these matrices represents its own unique. It regulates unique set of genes or downregulates unique set of genes, and some of these are closer to the patient, and some of these are distant from the patient. So for example, you have the Gefolger system is closer to the patient, and the RGD system is closer to the patient compared to the REDB-based system. Even when you compare these at the gene expression level, for the gene expression level analysis, you see there are distinct features that are showing up for RGD condition compared to the Gefolger condition and the REDB condition. So what we have tried to understand is that by exposing these tumors to different microenvironments, how is the gene network changing, and what are the distinct pathways that are changing? So you can find that information in this paper. In the interest of time, I won't be able to go through all of that. But in the last two slides, I want to wrap up by saying, I mentioned in the beginning that we use biomaterials to build technologies, but then we want to understand mechanism, and I showed you some of that mechanism now. But now we want to get into the therapy side of things. So one of the things that is important is that EZH2 inhibitors are now becoming or in clinical trials for several tumors, and we wanted to see whether inhibition of EZH2 using small molecule inhibitor of EZH2 has an impact on these tumors, and whether the matrix plays a role. And when you grow these tumors in matrigel versus designer matrix and hit it with the EZH2 inhibitors, it seems to be working very well in Gefolger collagen mimic or REDB mimic, but in RGD system, there is no difference between untreated versus treatment group. So it seems like RGD induces a distinct pathway response that causes resistance to these EZH2 tumors. And if you go back to this, indeed, RGD is upregulating a very distinct program over here from the gene expression analysis compared to the Gefolger and the REDB system. So it correlates very well in terms of why this might be happening with this. But the challenge with the EZH2 is that reported in the literature in some of our recent studies published in Blood Advances is that it is very good at suppressing the response, but eventually the response disappears and the tumor can bounce back. So there is a critical need for identifying combination delivery system, combination therapy system with these EZH2. So in this paper that was published earlier this year, we ended up identifying, using the sequencing analysis and some other analysis, a newer pathway called dopamine receptor D2 pathway, DRD2, and when we use that as a combination with EZH2, so for example, here is matrigel that you are seeing the response, and when you use RGD, which was showing a resistance over here, but when you use it with the combination, this is a very isolated piece of data. So full data, I recommend you to take a look at the manuscript, which with different controls. But when you take the RGD or the Gefolger group, in both cases, the combination kills the cells more effectively as compared to EZH2 alone group, where the RGD group doesn't show the killing response over here. So in other words, you can overcome some of these limitations imposed by cellular matrix by combining EZH2 with DRD2 inhibitor called ON201 and overcome that response. And as you can see, you have very much smaller volume of these tumors grown over a period of 40 days, 35 days compared to the control seen over here. So we believe that this combination might be of great interest moving forward. We're discussing this further, and either ON201 DRD2 inhibitor alone or in combination with EZH2 could be the potential next therapy for these neuroendocrine prostate cancers, for which there are really no treatment options at this point. So with that, I would like to end by saying that thanks to my entire team, this work was led by many of the people that have shown over here. I've been very lucky to have these outstanding trainees over my throughout my career. I showed data from many of these trainees, including my first PhD student, Alberto Perwada, over here, Chef Shah, and Liana Kramer's work was shown with the lipid nanoparticle system, and Matt Mascara did the prostate cancer organoid system as well as the gut microbiome system. And my collaborators, highlighted in yellow, who have contributed to a large number of this work that I've shown, and my funding agencies, without whom I would not be able to do this. A very exciting side of this is that we're now taking all of this and the mouse lymph node data that I showed, and we're building these human platforms, such as human tonsil organoid platforms, by using some similar approaches. And hopefully in the near future, I will be able to share some of that information. Thank you. Happy to answer any questions.
Thank you for this fascinating talk. Ankur, appreciate it. Many questions came in. One of them is, in your nano-vaccine study, do you consider creating gut-specific TLR5 knockout mice models to specifically study gut microbiome effect? I'm curious if whole animal TLR5 knockout would lead to other complicated effects.
Right, it's a great question. So, if I understood this the way it was posed, you can use the TLR5 knockout mice to study a very specific question that is related to the development of gut-induced microbiome disruption, gut-induced metabolic syndrome under the effect of TLR5 knockout. It's not identical to using an inflammatory bowel disease system. For that, you will use something else. If it's at the mouse level, you can use the DSS mouse model or some other genetically engineered mouse model. You can also, so in that paper, we use antibiotic treatment as an alternative to disrupt the gut microbiome. We saw similar results with PLG and vaccine not working over there. So there are many ways for you to study the gut microbiome condition. The TLR5 knockout does come with the notion that it will induce type 2 diabetes and systemic inflammation, which you can eliminate by using other models of disrupting gut microbiome.
Thank you. Another question is, how do you implement HLA and account for HLA's tremendous polymorphism toward understanding and modeling the immune decision-making process, both for cellular and humoral responses?
Sorry, can you repeat that question?
Sure. How do you implement HLA and account for HLA's tremendous polymorphism toward understanding and modeling the immune decision-making process, both for cellular and humoral responses?
Right. So that's a great question. So we haven't matched the HLA in the samples, but that's something that's a consideration as we are moving into the human system that we are doing. But that has never been our focus. From the mouse studies, most of the components that we are isolating are from the same strain of mice or from same mouse itself. And so we're less worried about that. When we're building a system that is more complex with multiple sources, that's where the HLA components are becoming more of a challenge. So, but that can be done if you look at some of the more recent studies on Mark Davis' group has published the human tonsils aggregates, and they have taken into consideration some of those aspects of that as well. Right.
Thank you. What are the markers or immunogenic epitopes, T cell or B cell specific ones that you use to characterize the specificity of the immune responses?
Right. So it depends on which population you are interested in. So for us, and it will change between mouse and the human. So for us, a general germinal center response is CD19 positive, GL7 positive, FAST positive response. You can also tie it up with various deeper analysis, such as light zone and dark zone markers, such as CXCR4 and CD86. And there's a series of markers that you can use to go one level deeper into analysis. Same way for the memory population. So we published a paper recently this year on how to derive memory population from these organoids, mouse organoids. And so their phenotype is different over there. And likewise, For the T cell population, you can look into generally the CD4 population, or if you want to go down the route of follicular T helper cells, then there are specific markers that you can define this. Now, going back to the question of if you want to do antigen-specific response, then you have to design very specific studies. So one way we did this was using a transgenic mouse where we used a B1-8 mouse that generates high affinity antibody responses. So we took the B cells from those and put them in the organoid and added an antigen called NP. It's a nitrophenol bound to albumin. And when you add that, it makes high affinity antibody against NP. So you can run flow cytometry to detect NP-specific antibody secreting cells by combining a bunch of these markers and by having tetramers or things like that that can bind to the antigen. So there are well-established assays and tools available at this point to take these cells out and analyze for those.
Thank you so much. How do you differentiate immunogen-specific immune reactions from auto or allo immune reactions?
We haven't done that. We have not. So the antigen that we add are targeted. So we add those and we look for a specific response against those. You can drive these reactions without any antigen. And in that case, I expect it to be forming polyclonal antibodies against random, a whole bunch of things that are there. So we haven't done an analysis of how much of this is autoantigen specific to be precise. What we did do was we have performed assays and we published this in 2018 Biomaterials paper, 2019 Biomaterials paper, where we did use the NP specific mouse model and an approach to use binding of the fast using anti-class antibody. And what it did was it depleted the non-specific B cells. So when you generate in the organoid, there are some cells that are going to be antigen specific, some cells are not going to be antigen specific. So we depleted the proportion of non-specific B cells and enriched the population of antigen specific B cells. That's how far we have gone in this system.
Thank you. What methods do you use to characterize antibody production? In particular, are you looking at the specific B cell clones and specific antibody epitopes that are generated? How about cross-reactivity?
So you can do all sort of simple to sophisticated analysis. We have done ELISAs. We're working with OSPOTs. We're also doing some more complex analysis with collaborators at Emory, where we are profiling for various different epitopes or cross-reactivity. So all the assays that are historically established over the years for fundamental immunology or, for example, vaccine or antibody responses can be applied to these, because the two ways to do this: one is extract the cells out by degrading the hydrogel using a collagenase, and second is by collecting the supernatant and running antibody analysis on it. So whatever is available in the field could be applied. We have done all sort of either we have done all of those or we're in process of doing some of those.
Thank you. Great talk. It seems the hydrogel is not required to tonsil organoid formation from Mark Davis's tonsil organoid paper. What's your insight of building human tonsil organoid with hydrogels?
Right. So it's a very, very nicely done study from Mark Davis group. And what they do is they take human tonsils and they cluster them together, put them together in a transfer-like format. The culture can go, it doesn't have a extracellular matrix component in any of those, but it's a pool of things coming out from a lymphoid tissue. Now, the tonsil already has components. It's always exposed to your oral microbiome, right? So it has components that may be in different states. So it can potentially help with that process. But I know they have done very neat studies where they took out the B cells, they purified naive B cells, and then performed those experiments to show that this can happen. One of the challenges that I see in that system is that we don't know, or at least by looking at that data that's published in the Nature Medicine paper, that the response drops down after, I believe, day 12 in that. And so the longevity of the system is unknown. And I believe that by bringing in a two-dimensionality with a right choice of extracellular matrix and various other components of the lymph node, you can perhaps prolong the longevity of that system. But it's a great system.
Thank you. Thank you. How is the microbiome affecting immune responses? Is it a direct molecular mimicry or indirect cytokine microenvironment effect, or both?
That's a great question. So this is the way we understand is that there is an impact of metabolites that are being secreted in this space, and there's emerging data coming out about bile acids and specific metabolites that function either directly on the B cells or through an inflammasome signature to regulate the fate of these B cells that are there. Of course, there are conditions such as aging and other conditions in which you have a leaky gut, in which components of the microbiome can leak out into the system, and that can have a different response or an added level of response to the innate immune cells, which can then direct the fate of these antibody secreting cells.
Thank you so much. Well, it was a great talk. Appreciate your time, Ankur. Truly appreciate it.
Thank you. Bye.
Bye-bye.